Hello, this is a quick recap of inherited infectious diseases. The first lesson I think everyone's gone through, but it'd be handy if you had a quick recap. Um, this is a list of the learning outcomes that we've all been through. So this lesson comes up to 2.3. Okay, so we'll go through that. Everyone's done this already. If you remember, we just needed the characteristics of viruses, bacteria, and Protoctista. We've all done that, and we've all got examples, so we can just quickly move on for the next one. We then went in to talk about antibiotics, and we said that we use antibiotics. We've taken them from various organisms because they have originally produced chemicals that kill bacteria. So this penicillium mold produces penicillin, and now we use that as an antibiotic. This is very important for us to understand that antibiotics do not kill viruses, they only kill bacteria. We then went on and talked about the modes of action of antibiotics. So we said that some of them interfere with cell wall synthesis, some interfere with cell membrane synthesis, some of those interfere with synthesis of the bacterium's RNA or DNA, and some of them stop the bacteria producing ribosomes. So they are specific. These groups of antibiotics are actually specific, and they stop the bacteria doing various biochemical processes. We then talked about antibiotic resistance, and if you remember, we had a look at this TED Talk video that was very good. You can watch that again just by clicking the link to talk about antibiotic resistance. We then had a look at the methods that bacteria are able to use to confer antibiotic resistance to other bacteria. So if you remember, we said that these bacteria have plasmids, which are extra genomic pieces of DNA, little circles of DNA that may have a gene in it for antibiotic resistance. And they're able to pass it to a bacterium that doesn't actually have it through these pili. All right, so right down the bottom, you end up with two bacteria, each of them now carrying the antibiotic resistant gene. We then had a look at some of the problems associated with antibiotics, because as you know, antibiotic resistant bacteria is a problem. What do we do once all the antibiotics don't work anymore? Before we get to that point, we need to make sure that we only prescribe antibiotics for bacterial diseases, not viral diseases, and to make sure that we take the full course of antibiotics. Don't stop taking the antibiotics when you start to feel ill. You've got to get through all of the bowl. Now, there's supposed to be a practical. Now, obviously, we can't do the practical part of the practical. We've written to Carver to say what they want us to do about this, and it may be that we either just disregard it, or as in just forget it and pretend it never happened, or they might want you to write something about it, as in a theoretical thing. But once Carver let us know what they want us to do, then I'll let you know. Okay. We went on to talk about Dr. Semmelweis, and it's kind of the history of science, really, of how we go from one step and then learn a bit more, really. We then have gone on to, and I can't remember. I think two of the groups have done this, and one group possibly hasn't. I can't remember. You have to talk about the method of transmission and treatment of a bacterial, a viral, and a that should actually be protoctistic disease. So I've chosen TB, HIV, malaria. Like I said before, if you've got your own favourite bacterial disease and you want to talk about that, please do, as long as it's accurate. If you've got your own favourite viral disease, you can do that. And if you have your own favourite protoctistin disease, you don't have to do TB, HIV, malaria. I suggest you do those because there's just an awful lot on the internet and in books about it. So it's probably the easiest ones to do. Infectious. So we're talking about infectious diseases here. So diseases that can infect one person and another. So all of those previous three are infectious. Of course, non-infectious diseases are things like, cancers and things like that. Right, tuberculosis then, quite apt at the minute with this COVID-19 thing going on, that it's spread by droplets, aerosols. So this person here has the bacterium tuberculosis, Mycobacterium tuberculosis. So when this person coughs and sneezes and whatever, then the bacterium is in these little salivary droplets. It then goes into the respiratory system of the recipient, and then it goes into their lungs, and then that's how that's transferred. That's one way that you can transfer TB. The other way is by drinking infected unpasteurized cow's milk. So I think I've done this other one. So pasteurization is where you run the milk through a high heat treatment to kill off bacteria, and that's what we buy in the shops, pasteurized milk. This thing I put in because everyone just assumes that TB is just a lung infection. It is a lung infection. You get these little tubercles there, and you can imagine they've got damage to the lungs. The person can't breathe properly, and that's not a good thing. But this is in there just to make us realise that tuberculosis, the bacterium, can go into other organs as well. It's not just the lungs. Diagnose it by X-rays and sputum samples. So that's how TB can be diagnosed. Okay, so hopefully you'll write something about those things in your assignment. HIV is our virus that we're going to look at. So as you know, viruses are not living; they're very, very small. The HIV virus structure, it's a protein coat with another protein coat on the inside, this little capsid, and inside you've got 2 lengths of RNA, nucleic acid, and then a couple of enzymes, reverse transcriptase enzymes, and this, like any virus, what it does is it goes into a host cell. They're obligate intracellular parasites. They go into the host cell. They then their RNA then breaks free. It's turned into DNA by the reverse transcriptase. The host cell, which happens to be a white blood cell in humans, then just does what this nucleic acid tells it to do, which is to make more HIV viruses. So it makes more HIV viruses, and then they burst out of the white blood cell, and the white blood cell is useless. And if enough of those white blood cells get damaged, then the person has no form of fighting other diseases, which is then when AIDS comes along. The spread of HIV, so we've got various lists here, and it's all well documented. Okay, in body fluids, it's this blood-to-blood transfer that's the main cause of spreading. It can be passed on from the mother to the child in breast milk. It can be passed on in infected blood products, but everything is just screened now, quite rightly, so that even if you do give blood, they're not going to use it if it's infected with HIV viruses. Controlling the spread again is very well documented. So what I want you to do is find some information on how you can control this, the spread of HIV, and then write about it. We then go on to malaria. There's our Protoctistin. This is our Plasmodium. All right, this is the little organism that causes malaria. And then the transmission of malaria. We've all got some sort of idea that if this person here has the malarial parasite, a mosquito comes along, drinks the infected blood, takes up the Plasmodium in its body, whizzes off to an uninfected person, drinks their blood, and then transfer the Plasmodium into this new person who's now infected, and so on, and that's how it goes. And I think I mentioned about the fact that this is in mainly tropical areas because that's where the mosquitoes kind of live. But with global warming and all that sort of stuff, they're actually moving north. They're probably moving south as well, but I haven't read anything about that. The mosquitoes are moving north, so who knows? Perhaps we'll actually get it in this country. Who knows? And then controlling malaria, you can control using drugs, obviously, so quinine and all that sort of stuff. I put these up here because they're sort of vaguely interesting. So here they're pouring, I think it's petroleum, onto these stagnant bits of water, because that's where the mosquito larvae live. That kills the mosquito larvae, so then you can't transfer it. The other way is this kind of physical barrier of a sleeping net. So you can impregnate the sleeping net with insecticide as well, which sort of puts the mosquitoes off. If you stop the mosquitoes biting the people, then hopefully that'll stop the spread of malaria, or at least decrease it. Anyway, that's a really quick whiz through that first couple of assessment criteria. If you have any questions or whatever, please feel free to email me or whamage me, and I will do my best to answer any questions you have. Lovely. Right, okay, I'll get on with the second one, and then I'll be able to send you the second video for the last assessment criterion. Thanks, bye.